Alright guys, so I took these notes, the faculty meeting today. Hopefully I just give you a breakdown of the types of things I think you need to focus your attention on for the upcoming exam. So I know the notes look a bit messy, but I'll kind of run you through and just listen to the things I'm talking about, and should give you a pretty good crash course in this stuff. So first things first, let's talk about DNA. I don't know why first things first, just because this is the first page that I pulled up. But anyways, we talked a lot about DNA, transcription, translation, and essentially comparing that to what we've learned about DNA so far, right? We know that we double the amount of DNA that we need during cell division in the S phase of interphase. Hopefully you guys know that. And that's when we make this stuff called complementary DNA, or cDNA. So a couple things you need to remember about DNA, right? DNA, it's the same in all of your cell types. That's kind of surprising to some people, right? Whether it be blood, muscle, it's still the same type of DNA. The difference is what genes are being transcribed and turned into proteins for that particular cell type to become what it is, right? So it's the same in all cells, right? Remember, this isn't every living creature, right? Plants, animals, it's all the same. We eat this stuff. If you can remember when we talked about, I think it was in the biotechnology section, when we discussed genetically engineered food products and so forth, which we'll talk about in a little more detail here in a little bit. We eat DNA all the time. How that affects our body, we're not totally sure, but it really wouldn't make sense that it does anything super detrimental to us, right? Because our body just breaks this stuff down, right? So it just says eat this stuff, okay? So DNA replication, that occurs in the S phase of interphase, right? And it uses the enzyme DNA polymerase. These are important because when we compare what we know about DNA so far, what we learned early in the semester, to the things that we're learning about DNA in relation to how we take the information on it and turn it into proteins, we need to compare it to the essentially single-strand version that we call RNA, right? Now remember some of the differences between DNA and RNA. DNA is this double helix structure, right? It just looks like a figure 8. I know, but it's supposed to be some two-dimensional double helix. You get the idea. It's twisty, it turns, whereas RNA is just a single strand. We know that DNA has these nucleotide bases, right? These nucleic acids here, or these nitrogenous bases are in these nucleic acids. We have A, T, C, and G in DNA, where we have A, U, C, and G in RNA. Okay, so some basic comparisons there. The sugar is a little different in DNA. It's deoxyribose, whereas in RNA, it's just ribose. And remember that the ratios of these nitrogenous bases is always the same. We did that question in class where we said, if you have 30% thymine, then how much guanine do you have? And it's just pretty easy to figure out, right? If you know A and T match up, then you have 30% A as well, and 30 and 30 is 60. So if I were to ask you how much guanine we have in DNA, if we know we have 30% thymine, then you just take 30 and 30, 60, and then 60 from 100 is 40, and then 40 is how much we have for cytosine and guanine, and we know if we divide 40 by 2 to get specifically guanine, then that's just going to be 20%, right? So pretty simple stuff, keeping all of these things in mind. Now, further differences with RNA and DNA, there are a number of types of RNAs, right? We have ribosomal RNA, we have transfer RNA, and we have this RNA that serves as a messenger, right? So let's talk about the differences in the way we use those, okay? So if you look at, let's say we're looking at a DNA template here. It just says template in the nucleus, right? If we're looking at some DNA template and we just randomly write down some bases, let's say we have the sequence, this 3 nitrogenous base sequence is known as a codon, right? Then right here, right, that matches up with the sequence on the DNA, then we can figure out what amino acids will be left behind using the methodology that we learn in class, right? So if we have some random sequence that maybe is going to turn into a gene to make some kind of a protein here, maybe GTA, TTA, for example, and we'll go back with these ones that I kind of scribbled out and wrote in bold over here in a moment. We're going to use this as a mutation. If if we have GTA and TTA, just some random gene sequence, we can figure out what messenger RNA sequence we'll get from that. And we can also use that fancy chart to figure out what amino acids will be left behind. But then you have these tRNAs. Go ahead and write this in there. These tRNAs, where your body uses to figure out what amino acid to be left behind. So let's say we have this sequence of bases here, GTA, TTA, and we want to figure out what your body is going to do to turn that into a protein. So first, what we have to do is we make a message that the other cellular machinery can use to essentially figure out what amino acids to leave behind. So we just match this up. We know we're using RNA, so we know we have no T's. Remember, that's the easiest way to double check, right? So we just G goes with C, T goes with A, but A goes with U, right? Because we're talking about messenger RNA, and then A, U, right here, right? So we have these transfer RNAs who are matching their anticodons with the codons on the messenger RNA in an effort to figure out what amino acids to be left behind in the next step, right? So first, we transcribed this gene, or essentially we're just looking at a small version of a gene, right? Or an abbreviated version. We're going to transcribe that into a messenger RNA, and then we're going to take that message and essentially we're going to decode it using the chart in our book that I'll put on your exam. Or your body is going to use transfer RNAs to translate that message into specific proteins, right? So if you look down here and you see these transfer RNAs, which are essentially just shuffling themselves into the ribosomes, right, which are made of ribosomal RNA. What they're doing is they're matching their anticodon with the codon sequence, which is just this set of 3 nitrogenous bases that we find on the messenger RNA. And they're matching that up. A goes with U, A goes with U goes with A. You get the idea, right? So we have that fancy chart though, right? And we can use this mRNA chart to essentially figure out that AAU translates to ASN, or that CAU translates to HIS. But your body doesn't have that fancy chart. So as I mentioned before, it uses these transfer RNAs that shuttle themselves into these ribosomes to essentially figure out that C goes with G, A goes with U goes with A, and GUA is the transfer RNA sequence that will leave behind HIS. But once again, I reiterate, CAU is what we use on the chart to figure out what amino acid will be left behind. So, I just randomly made up a scenario in which we had a possible mutation occurring here, right? So let's say we have a mutation occur. We have one of these nitrogenous bases mutate. We call that a point or substitution mutation because it doesn't shift anything except for that single base, right? So let's say this A switches to a G. Then we go through the entire sequence, and that changes our messenger RNA to AAC right here, right? And then our tRNA to UUG right here. And then we just look up and see what AAC is in our chart and see how it translates. And in this particular case, from this random mutation, we end up with the same amino acid sequences. And this is a really important concept because that means that we can still have a point mutation in our original DNA template strand that is transcribed and then translated into the exact same amino acid. So that's not going to change the protein structure once these amino acids fold into some three-dimensional protein to serve some specific function in our body. So in this particular instance, this is the only case that you can tell that we have a neutral mutation, right? If this were to switch to something else and it changes the protein sequence or the amino acid, then we wouldn't know if it was good or bad. And we'll talk about when we discuss selection later on this semester, how we can make proteins that may do a better job than they previously did, or how we can make proteins that do a worse job than they previously did. And that's a detrimental type of mutation, right? So we have transcription, making mRNA, and then translation, right? And that is essentially a breakdown of all the concepts that we learned in relation to DNA, how it differs from RNA, the process of transcription and translation, and how mutation affects this process.